Dear viewers, greetings. In this video, we are going to see about capillary electrophoresis. This video contains the following contents: introduction about capillary electrophoresis, followed by principle of capillary electrophoresis, types of capillary electrophoresis, instrumentation of capillary electrophoresis, applications of capillary electrophoresis, advantages of capillary electrophoresis, and finally limitations of capillary electrophoresis. Capillary electrophoresis. Capillary electrophoresis is a liquid-based separation technique that uses a capillary as a separation channel under the influence of an electric field. It is an analytical technique where the electroosmotic flow helps separately charged ions. The voltage applied for the capillary electrophoresis experiment are quite large, and it is often 10,000 volts to 20,000 volts. In 1980s, the capillary electrophoresis was emerged as a high efficiency technique with applications in various fields. Capillary electrophoresis technique is employed for the separation of minute quantities of substances within a short period of time, yielding exceptional resolution. Using this methodology, it is possible to effectively segregate a total of 18 amino acids within a time frame of 16 minutes. Capillary electrophoresis is a highly sensitive separation technique that has been primarily developed based on the concepts of high performance liquid chromatography, thin layer chromatography, and electrophoretic methods. The use of capillary electrophoresis has proven to be advantageous in the separation and analysis of biomolecules and compounds with lower molecular mass. Principle of capillary electrophoresis. In capillary electrophoresis, the electrophoretic separation occurs in a capillary tube of about 25 to 75 micrometer in diameter. A liquid sample containing molecules to be separated is first injected into the capillary either by applying pressure or high voltage power of about 10,000 volts to 20,000 volts. After injection, the electric field ionizes the molecules in the sample and forces them to migrate from one end of the capillary tube to the other end. At the detection window, an area in or outside of the capillary, the presence of the ions is detected using UV absorbance, fluorescence, laser-induced fluorescence, or electrochemistry. The differences in the electrical mobility of the ions are reflected in the time it takes to migrate to the detection window. This is termed as retention time or migration time and is shown as electropherogram. This figure shows the electropherogram from the sequencing of an unknown DNA fragment by automated capillary electrophoresis. Types of capillary electrophoresis. There are 6 types of capillary electrophoresis. They are capillary zone electrophoresis, capillary gel electrophoresis, micellar electrokinetic capillary chromatography, capillary electrochromatography, capillary isoelectric focusing, and finally capillary isotachophoresis. The first type of capillary electrophoresis is capillary zone electrophoresis. Capillary zone electrophoresis is a form of zone electrophoresis and is regarded as the basic mode of operation in capillary electrophoresis. In capillary zone electrophoresis, the capillary is filled with a solution buffer and molecules are separated in the free solution in the capillary. Ionized molecules migrate into distinct zones based on their electrical mobility, and the capillary zone electrophoresis is applicable in analyzing small to large molecules ranging from 2000 molecular weight to 1 lakh molecular weight. The capillary zone electrophoresis is a very efficient form of capillary electrophoresis. Capillary zone electrophoresis has been used to analyze amino acids, peptides, proteins, and other ionizable molecules, including the detection of drugs and drug metabolites in a forensic application. Capillary zone electrophoresis is also used in the separation and characterization of polar non-volatile compounds from environmental samples. The second type of capillary electrophoresis is capillary gel electrophoresis. Capillary gel electrophoresis is the embodiment of gel electrophoresis in capillary electrophoresis. In capillary gel electrophoresis, fluid cross-linking or linear polymers are filled into the capillary. Samples are injected using electrokinesis and separated by size and charge. While a support matrix is not necessary for capillary electrophoresis, the gel in capillary gel electrophoresis provides a molecular sieve which adds to the resolving power of the technique. Capillary gel electrophoresis has been applied to the analysis of nucleic acids, DNA fragments and protein. High resolution, sensitivity, reproducibility and automated nature of capillary gel electrophoresis has made it the technique of choice for Sanger's sequencing and for forensic DNA profiling. The third type of capillary electrophoresis is micellar electrokinetic capillary chromatography. Micellar electrokinetic capillary chromatography is a hybrid technique that merges electrophoresis and chromatography. Micellar electrokinetic capillary chromatography uses the surfactant in the running buffer above the critical micelle concentration. The added surfactant induces the formation of micelles in the electrophoresis system. The interaction between ionized molecules in the sample and micelles affects the separation time, enabling positively, negatively and neutrally charged ions to be eluted at different retention time. Micellar electrokinetic capillary chromatography can be used to separate and obtain all ion species. It has been used for a wide range of molecules including forensic analysis and elite compounds. The fourth type of capillary electrophoresis is capillary electrochromatography. Capillary electrochromatography combines capillary electrophoresis and chromatography, notably HPLC. A stationary phase coats the inner capillary wall, interacting with analytes similarly to traditional chromatography. This interaction combines with electrophoretic mobility and offers high resolution separations. Electroosmotic forces moves the mobile phase across the chromatographic bed, and the capillary electrochromatography separates amino acids, proteins, peptides and carbohydrates. Capillary electrophoresis is most widely utilized in pharmaceutical and polymer analysis industries to differentiate acidic and basic medicines. The fifth type of capillary electrophoresis is capillary isoelectric focusing. Capillary isoelectric focusing is the separation of amphoteric molecules such as peptides and proteins based on their isoelectric point using capillary electrophoresis. Here, the acidic buffer is placed at the anode and the basic buffer is placed at the cathode. The capillary is filled with a mixture of the running buffer and amphoteric molecules before an electric field is applied to establish a pH gradient in the capillary. Amphoteric molecules migrate through the gradient until they reach the point where the pH is equal to their isoelectric point. Detection is then performed when all molecules have successfully migrated. Capillary isoelectric focusing is typically used for the separation of amino acids, proteins, peptides and carbohydrates. The final and sixth capillary electrophoresis is capillary isotachophoresis. Capillary isotachophoresis technique was developed to separate the serum lipoproteins. Compared to other electrophoretic processes, it has negligible molecular sieve effects, requires no gel casting, applicable for whole serum and can easily differentiate lipoprotein subfractions. Here, the total serum lipoproteins are pre-strained in a free flow capillary system with an external diameter of 0.5 mm, having a discontinuous buffer system for 30 minutes at 4 degrees Celsius before starting the process. The lipoproteins are separated into high density lipoproteins and low density lipoproteins based on electrophoretic mobility. Capillary isotachophoresis is commonly used in clinical laboratories for determining the lipoprotein level in human serum. Instrumentation of capillary electrophoresis. The instrumentation of capillary electrophoresis contains power supply, buffer reservoirs, electrodes, capillary, optical viewing window, injection system, detector, thermostatic system, recorder, and computer or suitable integration. Power supply. Capillary electrophoresis requires a high voltage controllable direct current power supply. Buffer reservoirs. Capillary electrophoresis requires two identical buffer reservoirs filled with anodic and cathodic solutions. The stability of the electric field depends heavily on these buffer reservoirs. Electrodes. Capillary electrodes needs two electrodes, one is cathode, another one is anode. Two electrodes immersed in the buffer reservoir connected to the power supply. Capillary. Fused silica is used to construct the capillary that is utilized in capillary electrophoresis, and its diameter is typically less than 100 microns. The capillary functions as a route for the sample to pass through on their way to being separated. Optical viewing window. Capillary electrophoresis involves the utilization of an optical viewing window that is in proper alignment with the detector. The viewing window makes it possible to observe the separation process while it is taking place. Injection system. Capillary electrophoresis requires a suitable injection system for adding sample and buffer into the capillary. The method of injection can be automated for increased accuracy. Samples are often injected using one of the four methods: gravity or pressure or vacuum or electrokinetics. Detector. Conductimetric, fluorimetric, absorption spectrophotometry, that includes UV and visible, amperometric or mass spectroscopic detectors are all suitable for monitoring the amount of substances passing through the capillary at any given time in capillary electrophoresis. Thermostatic system. Capillary electrophoresis requires a thermostatic system for maintaining a constant temperature inside the capillary. Next, recorder. In capillary electrophoresis, the data collected during electrophoresis must be recorded for further analysis. The data must be recorded so that it can be analyzed later. Finally, computer or suitable integration. In addition, capillary electrophoresis requires a suitable integrator or computer for digital data conversion. It is easier to analyze and evaluate the digital data. Applications of capillary electrophoresis. Capillary electrophoresis is commonly applied in various fields. It includes analyzing foods and food contents, genetic differentiation, and clinical diagnosis. The following is the detailed explanation of the area of application of capillary electrophoresis. First, food technology. Capillary electrophoresis is applied to analyze different foods like beverages and fermented fruits to detect flavonoids, vitamins, carbohydrates, proteins, pigments, and color. Capillary electrophoresis is also applicable for detecting DNA and RNA from bacteria and viruses, which indicates contamination. The second application is DNA or RNA analysis. Capillary electrophoresis plays a major role in genomics and facilitating DNA sequencing, fragment analysis, and mutation detection. The third application is disease diagnosis. Lipid profile analysis using the capillary electrophoresis is critical for detecting cholesterol levels in clinical laboratories. Likewise, the human serum analysis of vitamins and minerals is performed using capillary electrophoresis. The fourth application is environmental monitoring. Capillary electrophoresis has been applied to track inorganic ions in river and water. Likewise, nanocapillary electrophoresis is applied to detect environmental pollutants at a nano level. The next fifth application is pharmaceutical laboratories. Capillary electrophoresis is used in the analysis of small molecules in drug during preparatory stages and standard solutions. The sixth application is proteomics. Capillary electrophoresis applied to the study of proteins and peptides and allows post-translational modifications to be uncovered and protein interactions to be identified. This information contributes to the discovery of potential drug targets and disease biomarkers. The seventh and final application is forensic science. Capillary electrophoresis facilitates forensic DNA analysis, which plays a vital role in criminal investigations and paternity testings. The technique's sensitivity enables the analysis of minute samples, often crucial in solving the cold cases. Advantages of capillary electrophoresis. The advantages of capillary electrophoresis are high efficient, high resolution, high sensitivity, versatility, and less time consuming. Finally, limitations of capillary electrophoresis. There are two limitations. The first limitation is changing capillaries is challenging. Due to the size of the capillaries, removing and replacing capillaries can be challenging at times. So, expertise is required for changing capillaries to the experiment. The second limitation is less robust. Small changes like the size of the capillaries and choice of the analytes can be crucial in capillary electrophoresis. So, capillary electrophoresis is less robust than other methods. Dear viewers, that's all about the capillary electrophoresis. Thank you for your support. Thank you.